Hello, everyone. My name is Bruno. I'm a plant biologist working on plant immunity, and I'm also a feature editor of Molecular Plant. Welcome to the video highlight of the first issue of Molecular Plant 2023. So this is a special issue for celebrating 15 years of publication of this journal. As you may know, Molecular Plant was published in 2008 and has served the science community for 15 years. In this special issue, a total of 16 papers were published, including 2 research articles, 2 resource articles, 11 reviews, and 1 opinion paper. So let's see the issue cover first. So the cover of this issue is dedicated to celebrate 15 years of publication by Molecular Plant. As one of the prime journals in plant science, Molecular Plant is dedicated to serve the science community by publishing novel and exciting findings with high significance in plant biology. So several representative covers published from this journal were put together to make up the number 15, which is surrounded by 6 other covers showing different plant species. So after seeing the covers, let me introduce you to some of the papers published in this issue. Photosynthesis is central to life on Earth, employing sunlight, water, and carbon dioxide to produce chemical energy and oxygen. One of the promising ways to increase crop yield is to enhance the light reaction of photosynthesis. Professor Dario Lester from Munich, Germany, contributed a critical review on this issue and discussed the strategy, controversies, and perspectives in enhancing the light reaction of photosynthesis. Cyclic electron transfer is also essential for photosynthesis, which is carried out by a series of membrane-associated proteins, such as the photosystem 1, cytochrome B6F, NADH dehydrogenases, and soluble electron carriers. Professor Mai Li and her colleagues from the Institute of Biophysics, Chinese Academy of Science, provided an up-to-date overview of the structures of these membrane complexes, and then discussed how they interact with mobile electron carriers in the cyclic electron transfer. And furthermore, Professor Alistair Alferny and colleagues reviewed the major progress on understanding the source-sink interaction in model plants under controlled growth conditions. They also analyzed the strategies used for increasing photosynthetic efficiency, and then discussed how the obtained knowledge can be translated into crops for optimized photosynthesis and yield improvement. Now, let's talk about plant genomics and crop research. This is a hot research topic in plant science from the past 2 decades. As we know, the draft genome of Arabidopsis thaliana and rice were published in 2000 and 2002, respectively. However, only one reference genome is not enough to understand the genetic variation within a plant species to harness for crop breeding. So from a population point of view, plant genomes are highly diverse, and the conserved and variable sequences collectively constitute the pangenome of a species. Professor Xiaoxu Han and colleagues provided a comprehensive review summarizing the development of the technologies used for constructing pangenomes and recent progress on plant pangenomic studies. They also discuss and showcase how pangenomics information can be used for both genetic studies and crop breeding. Crop improvement can be greatly accelerated by exploiting new methods such as genomic-based selection and prediction. Professor Hu Li and her colleagues from the Chinese Academy of Agricultural Science reported a deep learning method named Deep Neural Network Genomic Prediction, or DNNGP, for prediction of quantitative traits from multiomics data in the context of genomic selection in plants. This method performed as well as, or better than 5 other commonly used genomic prediction models when tested with 4 datasets in a broad range of prediction tasks. DNNGP also has several other advantages, making it a priming practical approach for wide application in crop improvement. The Green Revolution not only boosted the world's crop production, but also large-scale application of nitrogen fertilizers, which causes severe environmental issues. Improving crops' nitrogen use efficiency, or known as NUE, is crucial and urgent for sustainable agriculture. Professor Chang Chai-Chu and colleagues summarizes the approaches to genetically dissect NUE, modulating nitrogen utilization, as well as provided prospects and strategies for high NUE breeding. Now, let me introduce several papers on plant immunity. To resist pathogen interaction, plant has evolved two layers of immune system: pathogen-associated molecular pattern, known as PAM, triggered immunity, PTI, and effective triggered immunity, ETI, which is mediated by self-service localized pattern-triggered receptors, known as PRR, and intracellular immune receptors, known as NRR, respectively. Plant NRRs form these high-order protein complexes, called resistosome, in response to pathogen effectors. Professor Gigi Chai and his colleagues summarizes the recent findings concerning NRR resistosomes and discuss the biochemical and structural mechanisms of how immune signaling mediated by different NRR resistosomes converge on calcium pathway. They also discuss a potential role of calcium in the mutual potentiation of NRRs and PRR signaling. Salicylic acid is a defense hormone whose level increases rapidly after pathogen challenge and plays an important role on both local and systemic defense. While the core assay regulators and the key protein and assay biosynthesis has been well characterized, much remains unknown about the origin and evolution of this component. Professor Ki and his team identified 10 core protein families in assay signaling and biosynthesis across green plant lineage. They discovered that the key assay signaling receptors, the MPR proteins, originated in the most common ancestor of land plants, and also the full ICS-based assay synthesis pathway is also first assembled in the most common ancestor of land plants. So this study provided a significant insight into the early evolution and diversification of plant assay signaling and biosynthesis pathways, and also highlighted the role of assay in stress tolerance during plant terrestrialization. In another study, Professor Nem Hai Chua and his colleagues showed that the stability of salicylic acid receptors, NPR3, is enhanced by the increased assay level after pathogen infection. They found out that assay promotes the interaction between NPR3 and two deubiquitinases, UBP12 and UBP13. UBP12 and UBP13 removes ubiquitin from NPR3 and therefore stabilizing NPR3 to supply plant immunity. Jasmonic acid, or known as JA, is another plant hormone that's important for plant defense and growth. Professor Dao Xinxi and his team from Tsinghua University summarized the ligand receptor binding techniques used for the characterization of the phytohormone receptor interaction and introduced how the receptor, COI1, and active jasmonic acid molecules are identified. They then provided a comprehensive discussion about the recent advantage of the regulation of JA perception and its evolution. In addition to the papers highlighted in this special issue, there are also several other topics being covered, so please check the website to read these papers. Now introducing 4 review articles. The first one, Metabolomics, Centered Mining of Plant Metabolic Diversity and Function, Past Decades and Future Perspectives, by Professor Ji Liu and his team. The second one is Specialized Metabolites are Versatile Tools in Shaping Plant Microbe Association by Professor Powell Benarek and colleagues. And then the next one is the Regulation of Seed Storage Protein Synthesis in Monocots and Dicot Plants, a comparative review by Professor Yongru Wu and his team. And then the last one is the cellulose synthase in land plants by Professor Stefan Persson and colleagues. There's also an opinion paper, two-way communication, volatile emission and uptake occurred through the same barrier by Professor Natalia Vilda-River and colleagues. And then last but not least, there's a resource article titled The Protein-Protein Interaction Landscape of Transcription Factors During Genesium Development in Arabidopsis. Professor Stefan de Bolter's group reported a system biology study to identify the protein-protein interaction and dynamic spatial temporal network among transcription factors involved in the development of female reproductive organs in flowers. So this concludes the highlight of this issue. Thank you very much for listening.